All right, this is lab 8.1 analysis. I didn't show the inoculation of this. The other videos show how to inoculate the tubes and the media. So this here, the part A of lab 8.1 is starch hydrolysis media. So this is a starch plate, and here I have Bacillus subtilis, Escherichia coli, Pseudomonas aeruginosa. And with this test, we're testing to see if the bacteria produce an exoenzyme called amylase. So the enzyme would actually be secreted from the cell into the environment, and amylase breaks down starch molecules into monosaccharides, basically. The starch molecule is too large for the bacteria to actually transport into the cell, so it'll break it down and then move that into the cell. You can still see growth for all of them, but not all of them produced amylase. There would be monosaccharides in this media, which is kind of why you see, not so heavy growth. So to analyze and see this more visually, which one produced amylase, you flood the plate with grams iodine, and I'll take still images of this as well. But in the presence of iodine, starch will turn this like dark purple, almost black color. And where amylase is produced, there's a halo around the bacterial growth. And the glare of these lights here. It's hard to see with this, so I will take still images, but this one here, Bacillus subtilis, is the one that is producing this, has this halo indicating that it produced amylase. These other two have this purple color right up into the growth of that media, or the bacteria. So that's that first part of the lab there. Next part, part B, this is oxidation fermentation of glucose. So the OF media, this media was green to begin with, and I grew one with oxygen and one without. This is alkaligenase faecalis, and again I will post still images of this. But this one has like a blue kind of tint at the top, indicating something specific about this organism. In the reading for the entry to the lab, you can read what the blue color means. There's also pictures in the book on page 339, depending on which version of the book you're using. But this is the blue, and then without oxygen, there is no color change. So that was alkaligenase faecalis. Here we have E. coli, with and without oxygen. E. coli, both of those are now yellow. So that indicates what type of metabolism that organism is using. And then with oxygen, Pseudomonas aeruginosa. The media is kind of yellow. And again, it's hard to tell with this picture here, but this without oxygen, no oxygen, is still a green color. And again, I'll take stills of that. So Pseudomonas with oxygen is yellow. Without oxygen, the tube is still green. So this media allows us to tell if the bacteria is fermenting glucose or oxidizing glucose. Fermentation can occur with or without oxygen. The bacteria are not using it. Oxidation of glucose occurs with oxygen, so the bacteria is using oxygen as a terminal electron acceptor in the cell respiration. And then one of these organisms isn't using glucose at all, so you'll have to read that intro to lab 8 to determine which one is which. The next part is the nitrate reduction test. Here I have a spot plate, and I'm going to start with alkaligenase, and I'm going to pipette 1 ml into the spot plate. So alkylagenase on the left, keep that in mind. This nitrate reduction media is testing the bacteria's ability to use nitrate as an electron acceptor in an anaerobic respiration process. So following along with the little flow chart there, I have alkylagenase there. I'm going to add 2-3 drops of nitrate A reagent. 1, 2, 3. And then equal number of drops for nitrate B. And I have a toothpick somewhere to stir this. And we're looking for a color change to a red pinkish color after nitrate A and B. Put this under a white paper towel so you can see it better. Alkylagenase turn this pink color after nitrate A and B. So I don't have to do anything else for this organism. We have our result. The color change from this clear to red indicates that the organism may or may not have used nitrate. You'll have to read the lab to determine. You do the analysis. Next, I will use Pseudomonas. One mil in that too, the little well there. Same thing, 2-3 drops of nitrate A. Nitrate B. Give it a little stir. Wait a few moments. Not seeing any color change following that flow chart. Did it turn red? No. Add zinc dust. So we're going to add a small amount of zinc dust. Zinc dust will chemically reduce the nitrate if it's still there, and it would turn red if it was still there. Following that flow chart, is it red after zinc dust? No, this is not. That means it's a positive for bacterial reduction. Nitrate was reduced all the way to gas by the organism. The last one here is for E. coli. Again, 2 to 3 drops of nitrate A. And that one is turning out positive after nitrate A and B. So we had the alkylagenase, Pseudomonas, and E. coli. Next test is the oxidase test. Here we're going to simply add a couple drops of the oxidase reagent. We're looking for a color change to a pink maroon color within about 2 minutes. Usually it happens pretty quick. And I will take a still shot of this as well. It's very hard to see with this lack of focus on this camera. But I am seeing a pinkish color turn around the Pseudomonas, whereas the E. coli is remaining colorless. A little bit better there. See that pink color, that area. Whereas over here, there's no color change. And the last one, so E. coli, no change. Pseudomonas, pink color change. And the last one, we have the Catalase Test. We have Bacillus on the left, Streptococcus mutans on the right. I'm going to add a couple drops of hydrogen peroxide. And we're looking for bubbles indicating positive. It's hard to focus on these little plates. There we go. So you see the bubbles there on the left. Bacillus has bubbles. Streptococcus, no bubbles. Add a little bit more so you can see this better. Always fun to see the bubbles. Alright, so you'll fill out this lab report using this data. I'll take some still images of this, and then you can take the e-learning quiz associated with this, Lab 8.1.